All right, Amy, tell us about the work and what's important in using the materials you have to try to get regenerated bone.
Yeah, we're trying to develop spongy materials which have porous structure, and what is important for this spongy material is to have the right composition to grow bone tissue, to have the right porosity and pore size, and at the same time we're looking at making them multi-scale. It means that we would like to have small pores, large pores, and nanoparticles. Having nanoparticles make these structures to attach cells on them. I should restart. Yeah.
That's okay. Start again.
So
Having these.
Yeah, having these structures, having nanoscale features or nanoscale texture on these scaffolds help the cells attach to these scaffolds, and eventually once we implant them in the body, the scaffold can attract cells, and then we have regenerated bone. And the polymer we're using for making these scaffolds are biodegradable. It means that in the body, eventually they degrade over time, and then it is replaced with regenerated tissue. We are using modeling. It means that we're getting help from the Department of Statistics. So we're looking at the effect of several experimental factors. It is the size of macro channels or macropores that I mentioned. We're looking at the effect of composition because we're adding nanoparticles into the structure. How much nanoparticle do we need to add? And some other factors, what temperature do we need to use to create micropores at a size that helps with oxygen diffusion into these structures. And once we have all these factors optimized and we do optimization using a software, then we can take that structure, seed cells, we can have pre-osteoblastic cells or bone cells, or we can have human stem cells to grow on these scaffolds, and then they are implanted in the body. Right now we're using a bioreactor to generate an environment which is very close to our human body. So once they grow well inside the bioreactor, this gives us a good indication that eventually they can be used in animal trials. It could be implanted in mice or it could be implanted in rabbits, basically, to see how they are growing before they can be used for human clinical trials.
That's fabulous. Most people have no idea how many years it takes of research to know when you've got something that works. How long have you been working in this area?
We've been collaborating for over 7 years, I guess. And eventually, when we have these scaffolds work in animals, it doesn't mean that it is ready to be implanted in the body. Those polymers that are used in these scaffolds are FDA approved, but the device that we're making is not necessarily FDA approved because that is another step. So, but if we look at the market for skin regeneration, there are commercial products. For some cartilage regeneration, there are commercial products. For bone, we have demineralized bone, which are used as a scaffold. But having bone scaffold with cells inside and implanted in the body, it is another level of approval. It is not maybe approved yet, at least for our composition. That is another step that we need to take before it gets commercialized. So I would say maybe 10 years from now. So it's a long.
But that is your goal,
exactly.
Of course, right?
Hopefully.
Yeah. Okay. Thank you.